I'm Frank van Delft. I'm Professor for Structural Chemical Biology. Part of what we do here is the small research facility, historically called protein crystallography. What we do is collaborate on structure-based ligand discovery with cutting-edge approaches, including high-throughput crystallographic fragment screening and protein ligand structures, enabling gene products for crystallography using protein engineering, protein ligand cryo-electron microscopy, and microgram-scale array chemistry. Protein engineering is not a new thing. People have been doing it for many decades. What we are trying to do is really streamline and speed up the protein engineering to find the right crystal. We are working closely with the other small research facilities in the Center of Medicine's Discovery. What we're trying to offer to people working with us, collaborators, is the possibility to use very large numbers of compound protein structures to really accelerate the discovery. And what we've been building is high volume expression, rapid progression through purification into crystallization, so that in one go, we can, within a few weeks, explore dozens of variants straight into crystallization, evaluate if they have the right conformation for your discovery project, and then be ready to go. At the end of this process is our high-throughput crystallography infrastructure. And at the far end of that, we take anything that looks like a crystal and ship it off to Diamond Light Source to get quick readout of whether those are good crystals. And we then analyze them on batch to see if we have the right conformations. Diamond Light Source is the UK's national synchrotron, and it provides cutting-edge experiments to a wide range of researchers. The facility that I've built up there is called XChem, attached to a beamline I041. We help seed up to 40 or even 50 discovery projects every year with discovery groups from academia, industry, and other big partnerships. What Diamond offers via XChem is that users can come there and do an experiment that is difficult to do if you're on your own, but is very well streamlined at this facility. That within a week, one kind of screened about 1,000 fragments directly in crystal structures. And from this, you get a very rich picture of what the active site looks like and how you might develop compounds into that.
One of our collaborations is with Medicines for Malaria Venture and the Drug Discovery Unit in Dundee. We had a well-characterized malaria target that they already had chemical matter for, but they couldn't progress it to clinic. And the benefit of a technique like XChem is that we can go back and start from the scratch and identify chemophores that does not have the same problems that the original chemical series has, and progress that into viable binders, inhibitors, with the idea of getting to the drugs that we can actually take to clinic.
What we've been really cutting our teeth on recently is some very large antiviral discovery projects. The first big one was called the COVID Moonshot, which was a fragment screen in the heat of COVID against the main protease of the virus. That became spontaneous international collaboration. The project has kept going, and it is actually now declared as a preclinical candidate. But on the back of this, another big partnership grew. This big program is called AZAP, AI-driven structure-enabled antiviral platform. And here we've been developing the methodologies very aggressively for how we can take a list of targets and how we can work them up and get a set of results that are a platform for further drug discovery. As everyone knows, cryo-electron microscopy has had a resolution revolution over the last decade. So we are also now moving into the space for more complex systems.
We use cryo-EM and small molecule discovery to characterize how a particular compound or chemical interacts with its target, how it fits within a binding site and turns that protein on or off, what makes that molecule specific, what makes it hit only that target and not other off targets. And by using that information, we can design further generations of compounds that are more efficacious or maybe avoid off-target effects that would lead to side effects.
So if you have a gene where you have a very strong linkage to disease or other biological process which you want to interrogate or inhibit, come talk to us. We will help you get that gene enabled structurally, and we will help you get the information about protein-ligand interactions into your chemistry development pipeline and get to potency quickly so you can understand what's going on.